Hey everyone, we're pivoting away from metabolism finally and getting into all things cell division and nucleic acids and whatnot. So we're going to talk about the cell cycle, talk about tumor suppressor genes and oncogenes. We're going to talk about epigenetics, DNA replication, the whole mechanism, an emphasis on telomeres and how they're related to cancer and aging. We're going to talk about DNA damage, what's a point mutation versus frame shift mutation, DNA damage repair mechanisms. We have NER, BER, MMR, double strand break repair, transcription and transcriptional regulation with an emphasis on the lac operon. And finally, we'll close off with RNA interference and CRISPR. So for the cell cycle, we're just going to go through the different phases of the cell cycle with an emphasis on mitosis. What are cyclins and how do they relate to CDKs? And what for the G1 to S transition, the checkpoint, there's a lot of important proteins, retinoblastoma, P53, E2F that we should know about. And then we'll just sort of characterize tumor suppressors, oncogenes, and try to provide like a nice framework to a lens through which we can see and think about them. All right, here's our cell cycle. So G1, cells growing, cells functioning, nothing to write home about. S, we are replicating our DNA. G2, cell is growing, cell is functioning. And then mitosis, the cell is dividing, and mitosis is divided into prophase, metaphase, anaphase, telophase, and then cytokinesis, where we are cutting it in half. Sometimes people have an extra prophase, prometaphase, but I don't think that's so crazy important. Some cells will go into something called G0. So you might go into G0 if there's just not enough nutrients around, and it's just, maybe it's a bad time to, the same thing with humans, right? If a human female is going through a point of starvation, amenorrhea, she might stop ovulating, and it's just because it just doesn't make sense evolutionarily to get pregnant. If you're starving, you can't support a baby. Similar with division. If there's not enough nutrients, it's probably not a good time to divide. So it's going to hold off. But other cells are just permanently in this differentiated sort of quiescent state, something like a nerve cell or a muscle cell. Additionally, so some things don't really divide ever, and other cells might divide very quickly. A skin cell, a GI cell, any sort of cell that, an epithelial cell, something that's interacting with the environment, where there's lots of, mutagenic chemicals, reactive oxygen species, a lot of acid in our stomach. These cells are undergoing a lot of damage, so we have to replace them. So they or their progenitors might divide a lot. And you can cells are constantly picking up mutations and accumulating damage, but division puts an extra stress on the cell, especially during S phase, the copying mechanism might introduce a lot of errors there. So the more division you do, the more likely a cancer is to develop. So we might expect skin cancers and GI cancers and whatnot to be more common than a cancer that affects nerve cells, right? We know about brain cancer, lots of different cancers of the nervous system. They're actually usually going to be the accessory glial proteins, like in a oligodendrocyte or a Schwann cell. The actual neurons, because they're in G0, not dividing often, don't often get cancer. Of course, they can, but it's not as common. So the checkpoints, when we're looking at G1, we mentioned, want to make sure there's nutrients, want to make sure there's growth factors. The rest of the body and the rest of the cells are saying, yeah, we think it's a good time for you to go ahead and divide. And there's just no DNA damage, not a lot. If there is DNA damage, let's try to repair it. And if we can't repair it, maybe we should commit apoptosis and take ourselves out of the equation. After G2, we want to just make sure that, S phase went according to plan and that there were not a ton of mistakes. And then after M phase, let's make sure that all of the chromosomes separated, correctly. We don't have any sister chromatids still stuck together, things like that. Everything that's not mitosis is known as interphase. So mitosis. So let's talk about prophase and prometaphase. So our objective is to, we got 2n equals 46. So we've got 46 chromosomes, and we want to put, we want to split the sister chromatid. So half go into one cell and half go to the other cell. The most effective way to do that, just imagine DNA is like a ball of yarn like this. It's really difficult to separate the genetic information if it's in this yarn form. So you can imagine that one thing's going to happen is we're going to want to condense the chromosomes. It's really easy to separate, or easier to separate the DNA if it's condensed. Instead of yarn all over the place, we've condensed it into a little like spool of yarn. That makes it easier. Number two is we need something to pull the DNA, from to one side of the cell and then the sister chromatid to the other side of the cell. So we have these little centrosomes, which just have, little just centrioles, I would say. these are going to be our fishermen, which are casting, the lines to grab onto the chromosomes and pull them to the other side. So it would make sense that the chromosomes are condensing so that we can separate them easily. Two, the fisher people are appearing and they're starting to cast their lines. And then three, well, the DNA is stuck inside of the nucleus, and how the heck are you going to separate the genetic information if it's stuck inside of the nucleus? So we're going to go ahead and see that the nuclear envelope is dissolving. So we see that in both of these. Metaphase is just all of the chromosomes are nicely lined along the metaphase plate. We have our centrosomes, our fishermen on opposite sides, and they have successfully landed their mitotic spindle, which are microtubules, on the kinetochore, on the centromere of this of these chromosomes. Anaphase is just we're pulling apart, we're reeling the fission, and then telophase is really just the opposite of everything we saw in prophase and prometaphase. The chromosomes can start to uncondense. We want the nuclear envelope to reform, and the fishermen have done their job, so they can sort of go away. And we see all of that happening in this picture here. Additionally, though, we have cytokinesis, where a cleavage fur will develop, and we're going to have microfilaments sort of like choke off the cell so that it becomes, if you were going to like cut some, let's say you're making bread and you want to cut the dough with like a sharp wire. That's effectively what's happening here, and so we're going to split the cell in two. So pushing the cell cycle forward, we have these things called cyclins, and cyclins cycle. So we have D, E, A, B in that order from G1 all the way to M. And these are going to correspond with the cyclin-dependent kinases 4 and 6 and 2 and 1. So let's just look at G1. It's going to start to be transcribed and translated, and we see that the levels are going to go up, 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 and they peak right towards the end of G1. Interesting. They're peaking towards the end of G1, and that's because they're combining with CDK4. CDK4 is cyclin-dependent kinase 4. So when it's paired with its partner, they can now try and push the cell cycle forward. We then see similarly E accumulating. E is going to accumulate with 6. And together, the complex is going to try to push the cell cycle forward. And then once they've done their job and they've transitioned us into S phase, they can start to be degraded. Now it's job for cyclin A to start accumulating, and it peaks right at the end of S. And you know what? That's because it's going to team up with CDK2, and it's going to push us into G2. And then we're going to see the same thing with B, pairing up with CDK1 and pushing us into the M phase. And now a specific example in something we're actually going to go into more detail on is we might see like cyclin D in CDK4, and something that we're seeing happening as G1 progresses is we have this retinoblastoma protein. And notice that it's getting progressively more and more and more phosphorylated. And once it is fully phosphorylated, then we are allowed to proceed from G1 into S. So the CDKs of G1, it's important that they phosphorylate RB for us to get transition into S phase. But you know what? If there's damage, we might not want to go through with division. One of our checkpoints isn't met. And that's where we're going to have something like the P16 protein or other P proteins inhibiting the CDKs, because they're, on the one hand you can think of them as wet blankets, but on the other hand they're we need them, right? They're saying, hey, like maybe it's a bad idea to divide if not everything's perfectly going according to plan. So this is actually an entire mechanistic scheme that we should be familiar with. So this I think is very high yield to understand inside and out. So E2F right here wants to push the cell cycle forward, right? It is pro-division. Retinoblastoma here is a tumor suppressor. It is inhibiting E2F. That's its natural state of affair. So here it's inhibited by RB. But once we have the, CD, we have cyclin D and cyclin E pairing up with CDK4 and CDK6, those can come and can phosphorylate a retinoblastoma. And when it's phosphorylated, it is now inhibited, it's turned off. And you know what? That now means E2F is free to push us, yay, cell division, let's enter into S phase. But what if we were not ready to go into S phase? What if something's wrong? What if there's not enough growth signals or not enough nutrients, or we detect damage with the DNA? Well, then all of the P53s and the P proteins can come along and facilitate the CDK cycling complexes so that they do not phosphorylate E2F and it stays phosphorylate RB, so it stays inhibiting E2F so that we do not have cell division. So just to kind of summarize that, think of the P proteins as inhibiting division, the CDK cycling complex is prodivision, retinoblastoma as inhibiting division, and E2F as prodivision. And this kind of segues nicely into a tumor suppressor and a proto-oncogene. So some cells are anti-division and some cells are pro-division. You can think of these respectively as brakes and accelerators, right? When the accelerator's on, our car's going really fast, that is the cell cycle divide, going really quickly. Divide. And if the brakes are pressed, we're just not really going anywhere. We're not going to divide much. Now, if we have a defective brake, well, what happens if your car is a defective brake? You're going to go really, really fast because you can't slow down. So if we have a defective tumor suppressor, that could lead to cancer. Or if we had a hyperfunctional gene that's pro-division, that'd be like having the accelerator jammed on. That is also going to be pro-cancer. So we have tumor suppressors then and oncogenes. So very simplified, but we can think of as tumor suppressors as carrying less weight. So you just need one oncogene and it can lead to cancer. But again, super simplified, but one oncogene theoretically could lead to cancer, while you would usually want two tumor suppressors would have to be out of the equation to push towards cancer. This is known as the two-hit hypothesis. So we can kind of see this here. Imagine you have a car and there's, for whatever reason, a weird car, there's two brakes and two gas pedals. So what happens if just one of the brakes is fine? Well, if one of the brakes is fine, I'll just use this brake. I don't need that second brake. I kind of have a backup brake, which is nice. So you're good, no cancer, because you just typically need one brake. All right, what if both brakes are gone? Well, both brakes are gone, my car can't slow down, it's going to speed up, and that's more division. That's cancer. All right, what if I just have one of the gas pedals jammed in the on position? Well, if just one gas pedal is jammed in the on position, the car's going forward. It doesn't matter that I have a backup gas pedal. It's just irrelevant. Maybe even with the two brakes, the accelerator is jammed on, your car's probably going to go forward. So this would lead to cancer. Some people are born with one mutant tumor suppressor. So all of their cells have one of the brakes missing. And you can think of this as like flying without a co-pilot, right? If you have two pilots flying the plane and one of them passes out, well, you got a backup pilot, you're okay. But here we have no co-pilot. So you've got 40 trillion cells as an adult that divide, they're always dividing. It just takes one of those cells to get, so normally you're fine, right? But it just takes one of those cells to have a mutation. And you know what? Now you have two tumor suppressors missing, and that particular cell can lead to cancer. The P proteins are therefore tumor suppressors. Here we have gas pedals should say oncogene. Oncogenes are the gas pedal jammed on, and another tumor suppressor, and again oncogene. P53, if this is defective, this is a tumor suppressor. If we have both of them out of commission, or if someone's born with a leaf many, right? They have one defective P53. Well, it's just dumb luck. Eventually, they're going to get another P53 mutation, and this is such an important tumor suppressor that as they grow up, they're going to develop cancers all throughout their body. Same thing with retinoblastoma. This is an important tumor suppressor that we just saw. And you're born, you don't have cancer, but it's just a matter of time. At a very early age, you're just dumb luck, usually in one of your eyes, your retina, hence the name, or your bone as well. So you should know that it's the bone and the retina. You might get another mutation there, and that could lead to cancer. You see something called leukocoria. So normally, if you shine a flash with a camera into somebody's eyes, you get something called the red reflex. So this is normal. You get kind of this like red reflection. But if you do it and you get leukocoria, leuko means white, well, that's just because you're hitting a big tumor and it refracts the light a little bit differently. Another simplified rule too, just didn't know where to put this, but if we're thinking about dominant mutations versus recessive mutations, dominant mutations typically are when you're looking at something that's structural. So here we're seeing collagen, fibrillin, myosins and sarcomeres. That's a structural thing. Huntington is not necessarily structural when it's wild type, but the whole point of the Huntington disease phenotype is that these proteins are going to start to aggregate, and this aggregation is a structural thing. So these are all dominant mutations. Recessive is, again, where we have this idea of a backup. So for albinism, tyrosinase is necessary to convert dopa to melanin. And if you have one defective, well, that's okay, you got a backup tyronase. But if you have two defective, this is now going to lead to albinism. Phenylketonuria, we need phenylalanine hydroxylase to convert phenylalanine to tyronase. But you know what? We got a backup. But if you have two missing, now you got this. Same thing for this with gangliosides. CFTR gene is getting chloride from one side of the cell to the other side of the cell. If we have a defect, that's okay. We got a backup to get chlorine across. But so this is where it's two missing. In hemoglobin and sickle cell, well, that's okay. We're just transporting oxygen. But if we have one defective, okay, well, we can still transport oxygen. Okay, I guess. But if we have two defective, shit, now we're kind of screwed.